and also we saw understand. Oh, thank you.
I have the pleasure of introducing to you Dr. Gary Saunders, who is from University of New Brunswick in Fredericton. I've had the pleasure of being associated with Gary for a number of years. He is a very busy man who works on seaweeds all over the world, not just in this part, and I'm sure you will hear lots more about it. But I've been involved with him through the Sense of Marine Life particularly, and he has been lending his expertise to us many times. So thank you for that, and here it is to you.
Thank you very much. It's a great pleasure to be here. I haven't been down here since this new facility was put in place, and it's fantastic to see. It's really amazing, so I'm glad to be here. And I study seaweed, which is not something that a lot of people do. There might be 2 or 3 of us actually in the province that do, but it's a fascinating world that I really love working on. And we'll get the notion today that we're really going from the good old days to what may or may not be the good new days, depending on how you want to look at things. So a lot of people don't realize there's a lot of seaweed out there. They're in all marine environments. There's about 10,000 species of seaweed recognized on the planet. They live in the intertidal zone, which is something we have a lot of around here, down into the subtidal zone. So we were actually diving this morning here in the bay. And the deepest ones are found in the tropics, and the record is about 220 meters. That's a long way down. There's not a lot of light there, and these things still manage to do what seaweeds do, which is photosynthesize. What we're particularly interested in is the distribution, and there's different aspects to the distribution of seaweeds. There's a biogeographical component, so we feed very different types of seaweed in different parts of the world. There's different algae in the Arctic than there are in the tropics, then we get in the Atlantic, then we get in the Pacific. But there's also a vertical component, and if you go out on the beach, you'll see this thing we call the zonation of the intertidal zone. So up here we have our fucoids, and then we come into this zone of red algae here, and then finally down here we're getting into our kelps. So there's a very definite pattern to the way that these species are laid out on the different beaches in different parts of the world. And if you come from this part of the world, you're really adjusted to this. But when you go to the tropics, you don't see that because it's hot and the seaweeds don't grow on the rocks so much, and you really have to dive and do the kind of work we do. We get seaweeds living on rocks, we get seaweeds living on other seaweeds, we get them living on animals, we get them all over the place. And so here we have seaweed on seaweed. The funniest project I ever had was a guy sent me a bunch of crab antennae. And he wanted me to identify the seaweed that were growing on the crab antenna. And that was quite a fun little project because there were some pretty amazing things on these crab antennae. The other thing is you get a real community. So you get canopy type seaweeds, and if you move these things away and look underneath, you get these turfs underneath. So it's not just what you see on the surface, there's a lot of diversity hidden down underneath. And this is a whole range, there's probably about 12 different species right here of red and brown algae. So there's a lot of diversity underneath the surface canopy. And this structure is quite important because this gives us habitat for the various invert and fish larval stages. And so globally, algae are very important. They're responsible for about 50% of the carbon cycling on the planet. Unfortunately for me, most of that's phytoplankton. Seaweed don't contribute much to that. But seaweed are very, very important for this habitat. And so all your larval stages of your inverts, your lobsters, and your fishes are reliant on these seaweeds for their homes. This is another thing that most people are not aware of. You use seaweed every day of your life. It's in your foods, it's in your cosmetics, it's in your toothpaste. And so every night before you go to bed, you're getting a good dose of seaweed slapped across your teeth. So these are important organisms, and some of them are actually economically worth a lot of money. It's also a pretty major food crop in Asia. There are 3 major groups of seaweeds. There are the green algae, which are green, fortunately. The brown algae, which are brown, and then there's a group that I love the most, which are called the red algae. And the red algae are very complex because they have green, red, and blue pigments in them. And you can get different concentrations of these pigments. So when you start looking at red algae, they can be red, they can be dark red, they can be black, they can be yellow, they can be brown, and they can even be green. Now, these 3 groups of algae basically aren't even in the same kingdom. So animals are a kingdom, plants are a kingdom. These things are spread out over the tree of life. And sometimes when we pick up a red alga in the field, we don't even know if it's a red alga or a brown alga or a green alga just by looking at it. We can't even get it in the right kingdom sometimes when we're in the field. And that's part of what I want to talk about tonight. So the way we used to do this, and this is where we start our trip through the looking glass, is this historical approach to what we call biodiversity and biogeography, which is the kind of work that we do. And for hundreds of years, this was pretty much done in the classical botanical method. People would go out and collect plants, they would preserve these plants, and then they'd press them on herbaria sheets for storage in herbaria. This has been done. Darwin did it. Linnaeus did it. It's been going on for hundreds of years. And it's wonderful. You end up with these beautiful presses. You then spend a lot of time doing things like this, trying to identify these species based on their morphological attributes. So you set them down and you say it's got a stipe that's this long and this thick, its holdfast does this, the blade looks like this, and you pontificate a lot, and then eventually you put a name on it. And then you come up with these species lists, and then we get distributional limits, where these things grow and where they're from, which is the biogeographic component. And quite frankly, everyone was quite happy with that. It worked, and life was beautiful. The problem is with the seaweed is we were living in denial, because we really can't tell them apart. And here are some of the issues that we deal with. The first thing is that they have very complex life histories. Everyone thinks that life is like humans. Humans have more humans. Seaweeds don't do that. Seaweeds go through some very interesting and complex patterns. So for example, these kelps that you're familiar with here, these big brown blades, that's only one stage in the life history. There's another stage in the kelp life history that's this little microscopic filament. To try to link this to this using morphology is impossible. It was only when we started doing life history work that we started to realize that these things cycle through these very, very different stages, and we started to make those links. Because we have simple morphologies, we have high levels of what we call convergence. Convergence is when things that are independently evolving end up looking the same. And this happens a lot in the seaweed world. So here we have two things here that look very much the same, and they're not even in the same order. And you can't tell them apart in the field. Now, if I say they're not in the same order, that doesn't mean much to you. If I tell you a giraffe and a dog are not in the same order, that's what we're talking about here. Anyone in this room can look at a giraffe and a dog and tell them apart. But in the seaweed world, we can't do that. And finally, seaweeds are very plastic in response to the environment. It depends on where they're growing and how old they are. So these two things here are actually the same species. They are the same species, but it's a heck of a job in the field, unless you know that, to come to that conclusion. Because those are two very, very different looking plants. This is my favorite quote that I've ever tripped across in a phycology book in my life. This is actually the guide to the marine algae of California. This is the book that tells you how to identify seaweed from California. A good rule of thumb is that if specimens of red algae are sterile, so they're not reproductive, they should be discarded, since they would prove difficult to identify. There's advice. That's what we're dealing with. Those are the sorts of problems we have. So the world has changed in the seaweed, biodiversity, and biogeography. Today, we look directly at the DNA. We don't look at the morphology anymore, at least not first off. So everyone knows that DNA codes the blueprint of life as a series of 4 molecules. In eukaryotes, especially in the algae, they're found in 3 different compartments that we can look at this DNA from. But what's more important in my world is that DNA actually records the history of life. A lot of people don't know this about DNA, but it records the history of life, such that related species have similar DNA, and individuals of a species essentially have identical DNA. And we can use that knowledge to start to deal with the problems that we have in the seaweed world. We can actually assign specimens to genetic species groups. This is actually something that we've been doing in the seaweed world for, God, how old am I? 30 years, I would guess. And it's now become part of a much larger global initiative to use this sort of tool to identify organisms. So there's something called the International Barcode of Life. It's an international initiative to generate a short standardized DNA sequence for every eukaryotic species on the planet. This will provide the first accurate census of diversity on the planet. This will allow us to document species diversity at multiple geographical scales, which is what we're interested in. And the most important thing is we want to generate a database for rapid species identification. This was actually the launch of IBOL in Toronto in 2009. And those blues, reds, whites, and greens are representative of the 4 different nucleotides, and that's actually the DNA barcode, or the DNA sequence from a beaver, which is highly suitable considering that barcoding actually started in Canada. It's one of the great Canadian science success stories. And of course that means we've gone about tearing it all to pieces now, like we did with every other thing that we're good at. Okay, so what do? I have a collection. I don't know what it is. I grind it up. I get the DNA out. We use some tricks to get that particular marker we're interested in, that particular bit of DNA. We sequence it. We run it through a search engine, and it brings up genetic matches for us. And so I no longer have to worry about what this looks like. I just have to get this sequence out, run it through this database, and it'll tell me what my species is. And this works for a lot of different things. So that's the idea, or the game that we're playing now. It's very, very useful and very helpful. It's not perfect. There are problems with all types of science. This has huge practical applications if you take it outside of the seaweed world. Food quality. There's been a lot of work done analyzing fish in restaurants and in markets, determining whether or not you're actually getting what you're buying or a cheaper substitute. And I'm here to tell you the results are a little worrying. What is that hot dog made from? You can answer those sorts of questions. You probably don't want to know. But there's been very, very serious studies done on herbal tea, herbal medicines, determining whether or not they have the herbs and the plants in them that are claimed to be in them on the label, or whether it's substitutes that have been used from related plants. And so these are going to be very useful tools for these sorts of things. You don't need the whole organism anymore. So for wildlife control and management, because all you need is a fish scale or a hair from a car floor. It has DNA on it. Meat sample from a freezer has DNA. So any bit of biological material you can now identify. You don't need to see the whole organism. Customs are going to love this stuff. Insect larvae in a wood shipment. You can figure out what type of insect the larva comes from. You can identify unknown plant material. Honest officers, just hemp, that sort of stuff. Does a product contain biological material from an endangered or protected species? Leather belts, shoes, boots, purses, all these sorts of things can be checked. We can look at pests in agriculture. In fields, there stands its timber, and obviously there are ramifications for health. Did you actually eat what you thought you ate, or did you eat something poisonous? There is actually a gyromitra that looks very much like this nice edible mushroom here that is quite poisonous. And so these are the sorts of things that we can do. And in my little world down here at the bottom, it greatly helps us with biodiversity and conservation studies because we can identify organisms. This is the catch. Someone has to build the library. If you have something you want to identify, and it isn't already included in the database, you're not going to get a match. That thing has to be collected by a professional. They have to do the taxonomic work on it to identify it. They have to do the molecular work. And they have to put that sequence into a database. And this is the absolute irony of the DNA barcoding movement. I don't know how many hundreds of thousands, millions of dollars I've been given to do molecular work to generate sequence data, but I cannot get a single penny to go out and collect material and identify material and do that side of the work that's so critically important for DNA barcoding. And so it's, I'm always a glass half full kind of guy, but literally the glass is half full here. And no one wants to fill up the other side. In doing this work, though, it's been, I'm having a little trouble with this. It's been quite exciting. We've been all over the place, as you'll see. We've been in nice warm places with the gloves off. I'm down in Australia where we do a lot of our work. And we've been in places where you wish you had more than your gloves on. And that's the northern tip of Baffin Island in September. And I'm going to tell you that that was chilly.